So this is my route to the Ragon Institute. I'm from Zambia. I started working as a technician at the University Teaching Hospital in Lusaka, Zambia. I got the opportunity to come to the US to pursue graduate studies. Initially, I was supposed to spend a year. I ended up staying here for 10 years. After I finished my PhD at Hopkins, I joined the Ragon Institute. And after 3 years of my postdoctoral fellowship, I got a faculty position, and I opted to go back to Africa and set up my lab there. So in South Africa, I work on the fresh cohort that Doug Kwan has just been talking about, and I'm more interested in these very early events that happen soon after HIV infection. Because of the way the fresh cohort is designed, we can answer these questions about the early immunological events that occur soon after HIV infection. So when we did this study, what we found was quite surprising in that the body tries to do everything it can to try and fend off the infection, whereby almost 80% of the circulating CD8 T cells are HIV specific. But this response is quite fragile and just dies because these cells are highly apoptotic. More recently, like Krista said, these individuals are placed on therapy very, very early, and you can see how dramatic the picture looks now, where you can see we dump big viremia by several logs, and the CD4 T cells are maintained, and these people do very, very well in terms of their health. And so if we're going to cure HIV, we have a better chance with these people because they even have a very small reservoir. But we have a problem because we know antiretroviral therapy is not a cure. Even if we start treating people early like we do in FRESH and continuously treating them very closely, when they miss their medication for some reason, the virus quickly comes back up again. So the question is, where is the virus coming from? If we can find out where the virus is and eliminate it, maybe we can cure HIV. And that's how I became interested in the lymph node, because us and others have shown that the virus tends to persist in the lymph node. It is one of the so-called sanctuary sites. So we are curious to find out in FRESH, where people are treated in phoebic stage 1, very small viral load to begin with and suppressed within days. And Krista continuously monitors them very closely for adherence. Does the virus still persist in the lymph node? So again, we're very privileged because some of these people, agreed to undergo excision of lymph node for research purposes. So we obtained a lymph node from this particular individual and looked for HIV. To our surprise, we could readily detect HIV foci in the lymph node. This is an histological staining, and green there is GAG-P24. So when we zoom in here, what we find is that this HIV GAG-P24, that's the green stuff you see, is within this microanatomical structure known as the germinal center. And the reason the virus is hiding there is because when we look for CD8 T cells that are supposed to be killing HIV, these are the red stained cells there. They are restricted from entering this zone. Here now I'm showing you HIV GAG-P24 in yellow and the germinal center in green. The CD8 T cells, the red cells that are the killer CD8 T cells, are everywhere but the germinal center. So if you're going to eliminate HIV, we have to figure out how to can get the CD8 T cells to get into GC. And that's where we are working in collaboration with our colleagues here. All this work we're doing it in South Africa. We have high-end equipment now to do these kinds of studies. We have very, very talented students. And we collaborate closely with Alex Shelik, who is developing a lot of high-end tools that can help us to really answer this question of eliminating HIV in lymphoid tissues, and hopefully we'll get a cure. And Alex will tell you more about his tools. Thank you.